imagine the brain as a sort of nationwide satellite TV system. Across this landscape, every neuron works like its own broadcasting station as it sends out very regulated signals to the other stations. What we'll do in today's video is we'll follow one message through this network so that we can understand how synaptic transmission actually works. First, not all stations can communicate the same way. Some of them are connected by the electrical synapses, which are like the hardwired fiber optic lines between two TV stations. These ones are the gap junctions, which are channels that connect the cytoplasm of both cells. Ions or small molecules move directly through and also flow in both directions. The characteristics of this form of communication are that they are typically very fast, almost instantaneous, and very reliable. Electrical synapses also allow for a broadcasting that is synchronous. A good example of this is called the inferior olive, which is when neurons synchronize through the junctions so precisely that when they fire, it's kind of like a coordinated national broadcast. Electrical synapses also show up early in a brain's development, which helps the networks grow and organize due to their synchronous activity. However, most communication in the mature brain actually uses chemical synapses. In our metaphor, these are represented by the satellite-based links. So instead of direct wires, stations will transmit the signals across open air. This is when the metaphor becomes more useful. So, inside each neuron, vesicles are like the small storage satellites that orbit close to the transmission plane. Inside of these vesicles are the neurotransmitters, or the broadcast packets. When the neuron fires an action potential, this is basically when the station decides to broadcast now. When the action potential reaches the axon terminal, it leads to voltage-gated calcium channels opening, allowing calcium ions to rush in. This is the go signal that allows vesicles to be pulled from storage onto the broadcasting platform. The mechanism that fuses the vesicles with the membrane relies on snare proteins. They're kind of like clamps and docking locks that make sure the satellite pod is secured to the transmission port before the contents are released. Once these snare complexes engage, then the vesicle membrane is able to merge with the terminal membrane, and the neurotransmitter packets are able to go into the synaptic cleft. The synaptic cleft is like the air between the stations. Neurons actually never touch, so the signal has to cross the gap. The neurotransmitter gets released into the space, and they can be thought of as the TV signals or the broadcasted message that is encoded. Each neurotransmitter travels across the cleft and binds to a receptor, which is like a satellite dish that is tuned to a specific frequency. If the dish doesn't match the frequency, then the broadcast can't be received. That's how specificity in the brain works. The glutamate can only bind to the glutamate receptors, and the GABA can only bind to the GABA receptors, and so on. Now, when the next station gets the signal, the next question is, what does the broadcast actually tell it to do? If we think of it with the metaphor for the FCC, and for anyone who isn't familiar, the FCC, which is the Federal Communications Commission, is the U.S. agency that regulates all the broadcast signals, so it gets to decide who gets a green light and who gets a not today. The excitatory neurotransmitters, like glutamate, are like the FCC green lights, which tell the receiving station that they are cleared to transmit and that they should prepare their programming. In neurons, this usually leads to a depolarization, or when the neuron has a voltage higher than it usually does so that it can reach the firing threshold. Inhibitory neurotransmitters, like GABA, are like the FCC restriction notices, which tell the receiving stations that they should not transmit and should instead decrease their power. This usually leads to hyperpolarization, which is when the neuron has a voltage lower than it usually does so that the probability that the neuron fires is decreased. There are also some more subtle regulatory mechanisms, like shunting inhibition, which is when inhibitory signals don't hyperpolarize the cell much, but open channels that take away from the depolarizing current, like poking holes in a power line that the transmitter is using. The signal basically leaks out before it can reach the broadcast center at the soma. The receptors usually go into two main categories. The first is the ionotropic receptor, which is an immediate response dish. When the neurotransmitter binds, the dish directly opens an ion channel. This means that it is fast, local, and short-lived. The second type is the metabotropic receptor, which works through G proteins, which is a slower but more versatile way of communicating. When the neurotransmitter binds to a metabotropic receptor, the receptor activates a G protein by swapping the GDP for a GTP. Then the G protein splits into two parts, which is the G-alpha GTP and the G-beta gamma. Each part can then go on to activate different effectors. There are two major pathways here. The first is the shortcut pathway, which is when the receptor activation leads to G protein activation, which then opens an ion channel directly. This happens, for example, in your heart. In your heart cells, when a muscarinic acetylcholine receptor releases G beta gamma, this component directly opens a potassium channel and slows the heart down. This is fast, but still slower than ionotropic signaling. Secondly, there is the second messenger pathway, which is the biochemical cascade that can drastically change the behavior of the receiving station. A good example of this is the beta-adrenergic receptor on the postsynaptic neuron. When norepinephrine binds to the metabotropic receptor, it activates the G protein, GS. Then, inside the neuron, GS turns on adenylyl cyclase, which is an enzyme that immediately starts converting ATP into cyclic AMP, which activates protein kinase A, or PKA. PKA moves to specific targets inside the neuron. In this case, it moves to a set of potassium channels, and after phosphorylating them, they open less often. The neuron now loses an aspect of what's stabilizing the membrane potential and becomes a bit more depolarized and easier to excite. The reason these cascades are so strong is because of amplification. One receptor can activate lots of G proteins, and each G protein activates an enzyme that can then make hundreds of second messengers. Each of those activates multiple kinases, and each kinase can modify many channels. That's how the brain is able to make so much change from such tiny signals. The effects of these cascades include changed excitability, neurotransmitter release, and ion channel density. These are all long-lasting effects that can greatly affect learning and memory. For example, in neurons, beta-adrenergic signaling allows for inhibition of certain potassium channels because the PKA phosphorylates calcium channels. This allows for a slight depolarization that means the neuron is even more excitable. So let's look at it as one process, in the big picture. When a neuron fires, it initiates a broadcast, allowing the calcium to flow in, leading to the vesicles fusing via snares. Then, the neurotransmitters can be sent into the synaptic cleft, and the next station receives the signal through its tuned satellite dishes. Depending on whether the broadcast is excitatory, inhibitory, or modulatory, the receiving station either prepares to broadcast its own message, quiet down, or cause slower biochemical changes that can change in its future responses. This entire process is how the brain is able to coordinate our perception, thoughts, memory, and movements. It's the largest and most complex broadcasting network ever built.